Welcome everyone. My name is Sarah Burris. I'm the scientific liaison here at FUJIFILM VisualSonics. We have a very exciting webinar today titled Photoacoustic Contrast Media and Translational Efforts. This webinar will be presented by Dr. Jesse Jokerst, who is an associate professor in the Department of Nanoengineering at the University of California, San Diego. Now, just a couple notes about this webinar. This webinar is being recorded and this recording will be made available in approximately a few days. All lines will be muted during the duration of the recording, so if you have a question, please use the Q&A button throughout the presentation. Questions will be answered at the end of the session, and today's webinar is expected to last somewhere between 45 to 50 minutes with another 10 to 15 minutes of questions afterwards. So as I mentioned, Dr. Jokerst is an associate professor in the Department of Nanoengineering at the University of California, San Diego. He graduated cum laude from Truman State University in 2003 with a Bachelor's of Science in Chemistry. He then completed a PhD in chemistry at the University of Texas in Austin in 2009. He then was a postdoc at Stanford in the Department of Radiology from 2009 to 2013. And then was an instructor in that same department from 2013 to 2015. Jesse started at UCSD in July of 2015 and has received multiple awards from the NIH, such as the K99, the Pathway to Independence Award, the NIH New Inventor Award, and the NSF Career Award. In addition, he was also named the Stanford Radiology Alumni of the Year. His group has developed novel materials and optical excitation sources that improve photoacoustic ultrasound for use in medicine. Today, he's going to describe his lab's efforts to improve contrast via the customization of contrast agents and photoacoustic imaging. So with that, I'll turn it over to Dr. Jokerst.
Great, thank you so much for the introduction. So it's exciting to talk to everyone. I have actually been using VisualSonics technology. I was just sitting here thinking I was originally using the 770 for about the last few months when that, before the 2100 came out. So it's been really exciting to watch the technology change over the years, and it's definitely had a really valuable impact on my career. So again, I'm a chemist, and so a lot of what we'll talk about today are contrast agents. The group has expanded over time. About a third of my students, I guess, are more in the hardware and image processing space, and then there's an additional translational thrust that we'll talk about briefly at the end that is relatively light on contrast agents. But today we'll spend a fair amount of time on this. So the basic outline is I'll talk a little bit about traditional ultrasound, so like a pulse echo experiment, particularly as it applies to cardiovascular stem cell therapy. And then I'll talk about photoacoustic ultrasound and talk about several examples there, including image-guided drug delivery in cancer, monitoring anticoagulation, oral health, and just teasers on a few other projects. So I originally got excited about ultrasound for this because of this figure. When I started at Stanford, I was doing Raman spectroscopy, and at the end of about 2 hours, I could map a square centimeter of tissue and go about 1 millimeter deep. And so that was obviously kind of frustrating. And one day I went downstairs and started using some of the ultrasound equipment, and I could image in real time, and I could go many, many tens of centimeters deep into tissue. And so this figure kind of nicely illustrates how ultrasound compares to some other imaging and molecular imaging modalities. So if you've had a CT or an MRI, you know it's more like a snapshot. And ultrasound is basically like a video because we're taking hundreds of frames per second. Ultrasound also has very good spatial resolution. So that's what I'm plotting on the y-axis, is the smallest feature size that can be resolved. And so while some, like super resolution microscopy, can have very great spatial resolution, it frequently can't go very deep. And so ultrasound can have good temporal and spatial resolution. And if as a function of the spatial resolution, of course, is then a function of the center frequency of the transducer. And that's one of the signature contributions of VisualSonics are these high center frequency transducers, 40 to 70 megahertz. And when we go to those higher frequencies, we can have spatial resolutions that are on tens of microns. So these are all some of the things we like about ultrasound. The good temporal spatial resolution. Ultrasound is by far the most universally used imaging modality, from research hospitals to private practice clinics, used in a variety of applications. But some of the limitations are the low contrast to background ratio. It also primarily remains an anatomical technique as opposed to a molecular imaging technique. And so I think this image kind of nicely illustrates some of the problems that happen when doing traditional ultrasound imaging. So here's a ovarian cancer mass. This is the urinary bladder. And you can see it can be quite difficult to see this, this mass relative to the background tissue. So there's been a lot of work in this space using microbubbles. And so people will make gaseous microbubbles, and these have been used for many years in Europe. They're used, they're less common in the United States. But if you can think about the interface between a gas and a liquid, that is going to have a lot of acoustic impedance mismatch, and there's going to be a lot of signal. And so these bubbles have a long history of use as a contrast agent. Some of the problems with them, though, is that they're relatively large. And so if we want to image a target inside of a cell or outside of the vasculature, that can be difficult. They're also, what do bubbles do? They pop, so they're relatively unstable. And there's also been some reluctance to inject these gaseous particles into subjects just because of safety concerns. And so a lot of what the initial projects my group did is to try to develop nanoparticles that could be used as contrast media, but at a log order smaller size. So rather than having these microbubbles that are basically the size of red blood cells, could we start to achieve ultrasound contrast at much smaller sizes? And so these particles would then be integrated into the sample and increase the acoustic impedance backscatter of that sample. And this is just an image to illustrate the diversity of applications. So while most people are, of course, familiar with obstetrics, this ultrasound is used in many applications, particularly in echocardiography. And so some of the initial projects were exactly in that space of developing a variety of different nanoparticle types. And this was led by Fong Chen, who was my first PhD student and is now a fellow at Stanford. And so we looked at a variety of materials, and I'm just going to kind of boil this down into the greatest hits. And as this technology is applied to stem cell imaging in the heart. So just briefly, stem cell therapy has been done in about 6,000 human subjects thus far. And when one looks at a meta-analysis, there is a small but statistically significant increase in the performance of the heart muscle as a result of that stem cell therapy. So the exact mechanism is not entirely clear, whether it's due to differentiation of those cells, the release of paracrine factors from the cells, recruitment of endogenous stem cells by these exogenous stem cells. Regardless, there is increased cardiac output. And so after an ischemic event or a heart attack, there's generally some sort of reperfusion event and then the stem cell therapy. But some of the limitations here are answering just basic questions, such as how many cells are there? Are the cells interacting with the surrounding tissue? Are the cells alive or dead? Where are the cells injected? And so those are all questions that imaging is ideally suited to answer. And while conventional trials have used MRI, we suggested that ultrasound might be a particularly valuable technique in stem cell therapy, particularly in terms of understanding the delivery event. And so the basic idea is that we could create some kind of porous nanoparticle that would have acoustic impedance with the incident ultrasound and create an echo to report the presence of this nanoparticle. This same nanoparticle, because it's porous, could be a reservoir for growth factors to increase the survival of these stem cells after they're implanted into the heart. So by definition, these cells are being implanted in very necrotic and hypoxic regions of the heart. So anything that we could do to increase their viability would be useful. And we could also then potentially embed additional imaging agents such as radionuclides, or more likely gadolinium for T1 weighted MRI. So after these supplementary elements are put into the particle, the growth factor, the gadolinium, these particles are then embedded into the stem cells ex vivo. So this is just done in a tissue culture hood as the cells are being expanded prior to implantation. The cells are then purified and quantified and implanted into the left ventricle with real-time imaging via ultrasound. So this is a video of this, actually the delivery event. So just to orient you here, this is the left ventricle, and this is a short axis view of the heart. So the apex of the heart is into the board and the aorta is coming out of the board, and we're about medial into the left ventricle. So this is the area of the heart muscle wall, of the left ventricle wall, that we're going to be treating with MSCs. These are mesenchymal stem cells. And then this is a catheter that's been placed into the left ventricle wall. And so when I hit play, you'll see the relatively low echogenic signal here, very low signal, but as the cells are delivered now, you can see this hyperechoic region, this area of bright signal indicating the presence of those stem cells. And you can also appreciate that integration into the left ventricle wall. And we, of course, characterized this extensively with histology. We looked at how the motion of these cells corresponds to the ECG signal below. And then we could also confirm that these cells were in the left ventricle wall with MRI. So this is a T1 weighted MRI of these mice. The lungs, here's the left ventricle, and this is pre-treatment and relatively low signal. But post-treatment, there's this really obvious increased signal due to these nanoparticle labeled stem cells. So more recent work has looked at for functional characterization. And so this is now a long axis view of both a healthy and diseased heart. So this area here is the apex, and there's the aorta. This is one of the atria. And on the left, this is a healthy mouse. And on the right, this is a diseased mouse that we have given it an artificial heart attack. And so when I hit play, you'll appreciate on the left the relative size of the left ventricle wall. You can appreciate the symmetrical contraction of the heart, and you can appreciate how much of this heart chamber is being evacuated. So this is called the ejection fraction, how much of the total blood in the left ventricle is being ejected. On the right, you'll appreciate rather clunky movement, there's relatively little movement here in the apex of the heart, and this heart is just not doing a very good job of delivering blood to the rest of the body. So this is a metric that the VisualSonics technology is very good at measuring, is this ejection fraction, what percentage of the blood is being ejected. And so that's what we're plotting here, is this left ventricle ejection fraction, and this was a recent and relatively complicated studies were 12 animals in each group, and we monitored these groups over time. So our negative control were just the healthy animals, no heart attack. The positive control were animals that we induced this heart attack and gave no treatment to. And so you can see this really obvious decrease in the left ventricle ejection fraction. When we gave mesenchymal stem cells alone, there was initially maybe a slight improvement, but no long-term improvement over time. And this is likely due to the hypoxia and the poor viability of these cells in vivo. However, when we labeled these mesenchymal stem cells with the particle that was delivering this insulin-like growth factor as a pro-survival agent, we see this obvious and statistically robust increase in the left ventricle ejection fraction out to 60 days. Okay, so this is just some of the work we've done in traditional kind of pulse echo B-mode ultrasound. Transitioning now to photoacoustics. So photoacoustics is essentially another way of increasing contrast. If we think about the using contrast media as one way to increase contrast in acoustic imaging, photoacoustics is another approach. And so I'm sure many of the listeners are quite familiar with this technology, but for those who aren't, I'll briefly just describe it as a correlate to traditional ultrasound, where traditional ultrasound is sound in and then an echo and sound back out. Whereas in photoacoustics, there's an optical pulse and sound back out. So this is light in, sound out. And so Alexander Graham Bell actually originally described some of the original observations. Was certainly not using this for photoacoustic imaging, but has a patent on a device called the photophone, which was an alternative to the telephone. And it didn't work as well basically as the telephone, but if you want to look up that patent, you can definitely find it, that used the photoacoustic effect to transmit information. So photoacoustic combines the temporal and spatial resolution of ultrasound with the contrast and spectral nature of optics. So what I mean here is I had that original slide comparing ultrasound to other imaging modalities and showed that it had very fast frame rates and very good spatial resolution. So we still have that from ultrasound. But we also have is the ability to increase the contrast of our specific target area versus the background. And we can do spectral imaging, which means I now can start to have multiple color channels of information, which is relatively difficult in conventional ultrasound. So the mechanism is based on thermal expansion. And so there's initially an incident light pulse. And so this is often a OPO laser, a tunable laser. And these photons are initially ballistic, but when they enter the tissue, of course, they're absorbed and they're scattered. One of the fantastic features about photoacoustic imaging is that I don't care how many times this photon has been scattered. I only care about when it is absorbed, because absorption is what creates the signal. So in a typical optical experiment, like optical microscopy, if I have a scattered photon, it's a dead photon. But that's not so in photoacoustic imaging, which is great for deeper tissue imaging. So once the photon is absorbed, there is a very local temperature rise, and this is a spatially confined thermal expansion event. The reason that this is spatially confined is because these light pulses are very, very short. So these are about 5 to 50 nanoseconds at 5 to 50 hertz. So if you think about a second being 10 to the 9th nanoseconds, and about 10 times within that second, you do a pulse of 5 nanoseconds. The vast majority of that second, there is no optical excitation. But they're very, very short optical pulses, and therefore this local temperature rise is spatially confined. So don't think about this as photothermal therapy. We're not heating the tissue. We're not, there's no bulk temperature change. However, there is a very short and similarly spatial confined thermal expansion event. And so this thermal expansion event creates a pressure wave which can back propagate out to these transducers, which can then be, can take that information and reconstruct the image here shown in gray, which corresponds to the original target tissue shown here in bluish purple. Okay, so again, the advantage is high contrast, high resolution. Also, I can excite at 650, 750, 850, 950, and start to get multiple color channels of information based on the incident optical pulse. So that's truly a key feature of this technology. The limitations, it's still difficult to get light through bones, so imaging through intact human skull is really difficult. There's also just fundamental depth limitations, so this is never, at least not in my hands, going to be a full body technique because there's just, even though I can use the diffuse photons, there's only so far that photons will diffuse through human tissue. It's also difficult, we still have to couple these ultrasound transducers to whatever we're imaging. So air can be a problem, bubbles can be a problem. So these are the pros and cons of this technique. So what's doing the absorbing? Well, there's a variety of endogenous materials that are really great optical absorbers. Perhaps the best is hemoglobin and deoxyhemoglobin. And so others would be in maybe phospholipids. But what's nice is that it's very easy to use these digital ocean spectra of hemoglobin and deoxyhemoglobin and use this as a 2-equation, 2 unknown system to quantitate much hemoglobin in any given voxel or area of the tissue is oxygenated. We can also create a variety of exogenous materials, small molecules, nanoparticles, proteins. Most things that absorb light are photoacoustically active. So to talk briefly about some of these exogenous materials, this was a recent project that we did specifically for intraperitoneal chemotherapy. My lab doesn't do a lot of cancer, but this was a thought about for a while to sort of improve intraperitoneal chemotherapy. So this is a fairly invasive but fairly effective technique at treating primarily ovarian cancer, but also a variety of metastatic cancers that are in the abdomen. So there's therapy that's essentially infused to bathe the entire peritoneum in this chemotherapy. The NCI has recommended it for about 5 years, but it's rarely used because of some very severe side effects. So nanocarriers can help as a way of regulating the dose of these materials into the peritoneum. But similar to other examples of nanocarriers, it's very difficult to understand the drug release from these nanocarriers. So this work is trying to answer the question, is the drug cargo inside of the nanoparticle or has it been released? And so there's a lot of imaging that is done on the actual nanoparticle carrier itself, but much less so on answering that question, how much of the drug has been released and how much has it been released in vivo. So what we used is the model drug paclitaxel. And this was work with in my lab primarily, where we labeled this paclitaxel molecule with methylene blue, a derivative of methylene blue, which is a FDA-approved small molecule, primarily used for lymph node resection. So what's interesting about this small molecule is that it has an oxidized state and a reduced state. And when it's reduced, it is optically transparent, it absorbs light, and therefore it creates photoacoustic signal. However, when it's oxidized, it's blue, it absorbs light, it creates photoacoustic signal. So what we did is created this drug dye conjugate, encapsulated it into a particle, and locked it in the reduced state. And as this is released then, is it releases from the nanoparticle, it oxidizes, becomes blue, and is easy to quantitate. And this was actually on the inside cover last month of Angewandte Chemie. So we could first sort of characterize the drug dye conjugate both in its oxidized and reduced form and show really obvious differences in the absorption spectra, and then also do photoacoustic imaging on the drug in the oxidized form, calculate detection limits in the millimolar range, ex vivo. We could then integrate these drug-dye conjugates into nanoparticles and similarly measure the absorbance spectrum of the drug-dye conjugate inside the nanoparticles. So as you might expect, putting this pendant onto the drug has an effect on its ability to retard cancer. So this is, I believe, HT29 cells, and we're looking at the effect of dose for these 3 different systems. So the paclitaxel alone has an IC50 of around 68 nanometers. So this concentration that kills half of the cells. We need a high concentration to do the same amount of activity when we add this ethylene blue group. However, this could still be controlled by just adjusting the number of nanoparticles we have. So this is now the IC50 of this nanoparticle encapsulated system. And so this number could be reduced by perhaps increasing the space length between the paclitaxel and the target. This is obviously going to have some steric effects on the activity of the drug. So perhaps adding some more ethylene glycol repeats here would allow us to have a lower IC50 of the drug-dye conjugate. Regardless, we could monitor the amount of released drug via the absorbance, and then we could also take these same supernatants and measure the photoacoustic signal. So what's happening here is periodically taking down the particles, removing the supernatant, and watching this blue supernatant come off the particles and characterizing it with both absorbance and photoacoustics. So this is in vivo data, and hopefully, if we look at the most extreme case, maybe the difference between 0 hours and 8 hours, you notice this really obvious increase in photoacoustics. Did a control experiment. There's no increase over time, but this is suggested release of conjugate from the particles, and this is now just a subcutaneous injection. So the black and white here is the photoacoustic, the black and white is the traditional pulse echo data, and the red pixels are the photoacoustic imaging. So we then evaluated the in vivo activity, and this is with bioluminescence metastatic cell line that is labeled with this bioluminescence reporter. So we're just using this bioluminescence to monitor the amount of tumor in these animals. So 5 animals in each group. So we originally injected the tumors into the peritoneum and then dosed every other day, and at the same time points did bioluminescence imaging to quantify the tumor dose. So if you look at this line, this is just the empty nanoparticle. You can see that the empty nanoparticle has very little effect and the tumors continue to grow. If we inject PBS, there's a similar experiment. The tumors just continue to grow. We could use a very high concentration of paclitaxel that completely, well, not or nearly completely retarded tumor growth, but these animals also had such severe side effects that they had to be sacrificed. What we did find, though, is that this combination of the nanoparticle encapsulated drug dye conjugate had a significant impact on the tumor growth, and so this decreased the tumor burden by at least 50%, which was still an improvement over the same concentration of drug injected without the nanoparticle. Okay. So moving on from cancer, this is a completely different project. This was primarily initiated by Junshin Wang, who is my second PhD student and is now on staff at Intel in Arizona. And so the goal here was to better manage heparin anticoagulation therapy. So there's 500 million doses of this stuff given every year. It's given to people primarily with blood clots. So deep vein thrombosis, extracorporeal membrane oxygenation, people having high-risk coronary surgery, people on bypass, people on kidney dialysis. Anytime there's a risk of a blood clot, people are frequently given heparin. Heparin's difficult to manage. So this is a cartoon of a typical time course of treatment where the activity goes up and down and up and down, and it takes a while to land the dose into the therapeutic window. If there's too much, you have a risk of hemorrhage and bleeding. If there's an underdose, you have a risk of clotting. And so the dose is controlled by a blood test known as the partial thromboplastin time or the active clotting time. We suggested that by using imaging and increasing the time of testing, we could very quickly titrate and keep the patient in this therapeutic window where the drug activity is both safe and effective. So the long-term vision is there would be some kind of real-time feedback via imaging that would feed into the IV infusion pump and control the dose of heparin that's going into the patient. And the basic discovery was that, again, our friend methylene blue, this FDA-approved dye, has a dose-dependent increase in signal when it interacts with heparin. And the mechanism is that there's a delocalized positive charge on blue, which is attracted to the sulfonated backbone of heparin. And when these methylene blue molecules interact with heparin, they dimerize or form higher order aggregates. And as a result of that aggregation, they have less interaction with the solvent and less fluorescence, and therefore their photoacoustic signal goes up. So this is a simple experiment where there's the same concentration of methylene blue in all of these tubes, but increasing concentrations of heparin. And so you can appreciate really obviously that the photoacoustic signal goes up as a function of the concentration of heparin. We could also further prove the mechanism with the known antagonist to heparin, which is protamine sulfate. And so this experiment has the same concentration of heparin and methylene blue, but increasing concentrations of protamine sulfate. So protamine sulfate is going to have an even higher affinity for heparin, and therefore this methylene blue becomes free and is back in solution, and the photoacoustic signal goes down. So what to do with it? Well, we're working on essentially using this for real-time sensing, coating this heparin-sensitive layer onto a catheter. So these people are these are sick people. They're going to have at least one IV port, if not multiple. And so, and heparin is already given intravenously. So imaging it or monitoring it intravenously is the logical next step. So we would make this catheter functionalized for heparin. This would be in the vein, and then there would be some kind of external transducer to monitor that activity. And so we have validated this is just the signal response of this first generation functionalized heparin catheter, and shown that it's responsive to different concentrations of heparin. And more recently, we've used an analog of methylene blue known as Nile blue. And this is work with Bill Penny at the UCSD VA hospital. And so we're not putting this catheter into people, but we are getting samples from the operating room in Dr. Penny's cardiology clinic. And so what we did here for, I believe this is 40 patients, we measured the activated clotting time, or at least we took the activated clotting time that Penny and his colleagues measured in the operating suite, and then compared that to the photoacoustic signal of this catheter and correlated the two, and showed this promising correlation between the two, suggesting that we're not just imaging activity, we're measuring function. We're measuring how much anticoagulation activity is there in this patient. And then we further quantitated the cumulative heparin dose with these two techniques. And this was just an important control to validate that the dose was correlating to our signal. So future work in this space is validating this with some animal models. Rabbits have a nice ear vein. So we need to be doing the imaging in vivo, and also moving away from these sorts of bulky transducers. What's nice about this approach is we're not really doing, we don't even need to do imaging. So a single element transducer would be just fine for measuring the signal because we don't need to create an image. So some sort of real-time sensing over that catheter. The last one that I'll briefly describe well, not briefly describe in detail, describe in detail is periodontal imaging. So this is primarily being led by Coleman Moore, who's a NSF fellow in the lab. And so I don't know how many of you have had this done, but this is a common procedure in North America called the periodontal probing. And so if you've been to the dentist and they lean you back and they say 3, 4, 4, 5, as they probe this little metal stick around your mouth, what they're taking is what's known as the periodontal probing measurement. So what they're doing is they're taking this metal stick and they're poking it in between your tooth and your gum, okay? If your gums are in good shape, this little metal stick should not go down very far. So those numbers they're reading off, 3, 3, 4, 4, those are how many millimeters deep it can go. However, if your gums are inflamed, the tooth and the gum do not have a lot of affinity for each other. Your tooth is loose, and therefore this little metal stick can go down a lot farther into this space between the tooth and your gum. This space is called the periodontal pocket or the sulcus. So you can imagine there's a number of problems with this. Patients don't like it. Technicians don't like it. Periodontists don't like it. Dentists don't like it because it's time consuming. It's very air prone. The force that is used, the angulation, the patient's hydration status. So the last time I had this done, I thought there's got to be a better way. And so the long-term vision here is some kind of oral rinse that would be administered, and then a customized transducer that would non-invasively extract these methods or these metrics of gingival health automatically from an image. So what is the contrast agent? Well, I was thinking about things that could quickly get IRB approval. And it turns out that these adorable little sea creatures, cephalopods and their friends, when they get stressed out, they emit a cloud of black ink. And that black ink is actually made of tiny little melanin nanoparticles. And so melanin is a strong optical absorber and has strong photoacoustic signal. But more importantly, squid ink pasta and squid ink is a very common ingredient in a variety of foods, and you may have had squid ink pasta. And so I thought, of all the things to tell the IRB that I'm going to put into people's mouths, something that's an ingredient in Mediterranean cooking might be a good one to propose. And so this is a swine model, and the black and white image again here is again the ultrasound. So these swine didn't take very good care of their teeth, and so there's a lot of background staining. This is the plaque and tartar that's on their teeth. I drew in PowerPoint here the gingival margin, and then after this oral rinse, after these squid ink particles are delivered, you see this really obvious increase in the photoacoustic signal in a different color channel in the sulcus. And so here we can measure this periodontal pocket at different sites non-invasively. So what's nice is that you get the entire contour of the pocket rather than a single point measurement that you might get with conventional probing. So the reason there's two different colors there is simply because of these two different absorption spectra. And so we're doing, again, ratiometric imaging at 680 and 800, and could use these two spectra to determine what signal belongs to which feature in this swine's mouth. So then we could look at a sagittal section here. And so here you can see hopefully this blue color of the stain, the probing depth here, there's the bone, there's the gingiva, there's the top of the tooth. This is the part that does the biting. So then we could look at these deep pockets through shallow pockets, and we could correlate this probing depth by imaging to the probing depth with a conventional periodontal probe and show this nice correlation between the two. Because we were using high frequency ultrasound, we could very precisely measure the thickness of the gingiva. This is also known as biotyping. And so it's very difficult to answer this question otherwise. It's basically just done based on observations, but this is a very precise way to measure how thick is the gingiva at any point and how does that thickness change throughout the gum flap. And we, again, correlated this to the closest alternative method, which is sort of calipers. And we showed really no bias between this new technique and the conventional technique. You might think, well, okay, that's all well and good, but I don't want my teeth to turn black after I have this imaging done. And so here's the baseline situation showing just the background plaque and tartar. So the particles are administered, and then the swine jaw was briefly rinsed with water. And so we show relatively little loss of these particles until there was a careful tooth brushing. And then when we brushed the jaw, all of this signal from the squidding particles quickly went away. So we could also, rather than using this exogenous material, we could also do endogenous imaging. I told you that using a 2-equation, 2-unknown system, we can quantitate how much of the hemoglobin is oxygenated at any point. And so that's also what we did to try to see if there's any value in periimplantitis. And periimplantitis is a very important and difficult situation to evaluate clinically. So this is, as it sounds, it's when there's an infection or an inflammation around an implant. And conventional dentistry uses x-rays, right? X-rays are great at imaging bone and hard tissue, but ultrasound is great at imaging soft tissue. So the question clinically is, do I need to remove this implant? And it frequently is not removed until there's very significant bone loss shown down here, at which point this bone, so much bone is lost that it can be difficult to ever replace this tooth. So what we want to do is use imaging to monitor periimplantitis and potentially guide treatment decisions. So just as a rough proof of principle, we took a rabbit model. So here, these are rabbit incisors. So you can see the bright area here corresponding to the surface of the tooth. And then this is the rabbit's gingiva. And we could change the oxygen tension that we were giving these animals from normal room air through 100% oxygen down to only 5% oxygen. And you can appreciate really obviously the signal changes that result from these different oxygen tensions. We could correlate that to pulse oximetry. So we could put a little, pulse oximeter on the rabbit's paw and show that we were indeed changing the oxygen tension in the animal. But then more importantly, this is a model of periodontal disease that we used a ligature that was soaked in some periodontal pathogens, tied around the tooth, and over time you can see this obvious inflammation. And so this obvious inflammation then could be confirmed with photoacoustic imaging, and we show this hypoxia in the inflamed tissue relative to normal tissue. So you might say, well, why would I ever need imaging? I can see this by eye. But that's true, but you can't appreciate the depth of involvement, and particularly the depth of involvement when it would be surrounding an implant. And so this is ongoing work and work that we're excited about. We have done some clinical translation of this. So this was, we've published one patient just as a proof of principle where we immobilized the subject's head and scanned this transducer across the mouth. And so because of the size of this transducer, we couldn't access the molars. We could only access the incisors and canines, but we could get these decent B-mode images of the incisors and the canines, both these in-fos images and these sagittal sections where you can appreciate there's the root of the tooth and there's the gum flap for all of these teeth. And then this is some of the probing depth measurements where we irrigated this pocket, again with the squid ink particles, and you see the obvious similarities between the swine tissue and this human subject. So this was exciting. So this was back in 2018. We had just launched a larger clinical trial before, and we got up to 5 human subjects before the coronavirus shut us down, but that continue whenever we reopen. We're excited about the reproducibility that we've showed earlier. So for one tooth, repeat the measurement 10 times or 5 times and show about 10% reproducibility. So this is larger work. There's no dental school at UCSD, so we're working with Keishi Chen, who's chair of perio at USC. We're also building customized transducers for access to molar farther back in the mouth. So where are we on time? Yeah, 5 minutes. So just a few other projects. This is worth Pranav Garamella and again Bill Penny on some wound imaging. So there's a number of types of chronic wounds. They're very expensive. They're very difficult to treat. So these are things like diabetic foot ulcers, pressure ulcers. These people get these open wounds and they tend to progress over time. It can lead to amputation. And so we thought, could we use imaging and photoacoustic imaging to help in the treatment of this? And so you might think, why would I ever need imaging to image something like that? Well, we wanted to diagnose before these are visible by eye. And so what we did in an animal model is look at multiple stages of pressure wounds and show that the photoacoustic signal intensity increases over time, but also the depth from the surface of the skin increases. So if you look at how far deeper into the tissue this photoacoustic signal is here, then over here, these were 2 imaging biomarkers that we could use to stage different ulcers. And then if you go and read this paper, you'll see that we could go and image animals that were pre-stage 1. So before there were even broken skin, we could start to detect these ulcers. There's also a potential argument one could make that using imaging could guide treatment decisions and evaluating treatment responses. And so this was another trial that we were up to subject number 4 that got stopped right before we could get much farther because of the coronavirus. I'll also just briefly mention oxidative stress imaging. So this was a project started by Taeho Kim, who was a postdoc in the group and is now on the faculty at Michigan State. And so what we did here is use these plasmonic particles, plasmonic gold, and when we coated a silver layer around them, they have a very obvious blue shift in their optical absorption. However, when the silver is oxidized back off, we recover this absorption. So if we're doing photoacoustic imaging at around 700 nanometers, this particle is on, this particle is off, and this particle is on again. So what's interesting about them is that we could use this oxidation, or we could control this oxidation by reactive oxygen species, such as hydrogen peroxide and peroxynitrate. And so there's this very dose-dependent signal change as a function of the quantity of these 2 reactive oxygen species. And so this photoacoustic signal is very linear with the quantity of these ROS species, but also it's very linear with the amount of silver ions that have been released. So there's a number of things one could do with this. One could use the silver ions as a therapy. One could also use this as an imaging agent to quantitate how many reactive oxygen species are present in tissue. And so reactive oxygen species are very difficult to measure. They're very short-lived species, generally at low concentration. So doing a blood draw, for example, and taking it to a lab would never work. So it's almost an experiment that has to be done by imaging. Finally, we've got a large project with Bill Vogt at the FDA working on building some standardization phantoms. So as this technology continues to mature, you'll see a number of pieces of equipment submitted for FDA clearance, but there's actually not a lot of truly well-characterized phantoms. Phantoms tend to be made in each lab, and so we're identifying materials that mimic both the optical and acoustic properties of tissue to help facilitate these standardization efforts. So with that, I'd like to thank everyone in the group, these sources of funding, again, VisualSonics for the invitation, you for your attention. I also am always looking for great people, smart, hardworking, clever, so feel free to reach out. And with that, we'll save some time for questions.
Great, thank you, Jesse, so much. That was an amazing talk. We'll just be waiting for some questions to filter in on the Q&A. We have several that have already come up. Jesse, can you tell us a little bit about how deep you can image with photoacoustics?
Yeah, so it really sort of depends on the tissue type. So, for example, if it's a particularly, for example, imaging through bone is more difficult. So for imaging through a mouse skull, generally 1 centimeter is about as much as we can hope for. If it's in sort of a more optically transparent tissue, people routinely go 4 and 5, and then there's occasional reports of up to 8 centimeters in the literature. It's also a function of your optical fluence, how much optical dose are you delivering, and how strong is the absorber. But 3 to 4 centimeters is a pretty good rule of thumb.
Yeah, great. Can you tell us a little bit about what the clearance of some of the contrast agents are, what their stability is like?
Sure, yeah. So the plasmonic materials tend to be slightly less stable. It turns out that, for example, on the gold nanorods, a nanorod-like shape is one of the least thermodynamically favored conformation. And so when you give this nanorod this extra energy in the form of an optical dose, it can frequently convert back to a sphere-like shape. Others, like other small molecules or other nanoparticle types like melanins, those are very, very stable with repeated optical excitation. In terms of clearance, yeah, kind of standard metrics. Small molecules tend to be cleared renally. The nanoparticles tend to be cleared hepatobiliarly.
Great, thank you. We have a question related to what is the estimated increase in temperature? And I assume they're talking about the thermoelastic expansion?
Yeah, I have never measured. I've tried it on my first few papers. This question would come up, and I've never been able to measure a temperature increase, a bulk temperature increase in the tissue. I'm sure someone has calculated theoretically the spatially confined temperature increase, but I don't know that off the top of my head.
Okay, thank you. You're definitely getting a lot of praise coming in over the Q&A session.
Sure.
Someone specifically says, I'm working with semiconducting nanoparticles for in vivo imaging. I saw very different photoacoustic amplitudes depending upon the envelope material. Do you know what the reason for that could be? Are there different thermo properties like specific heat capacities or thermal conductivity?
Yeah, so I don't have the full, I didn't show the full photoacoustic equation, but photoacoustic signal is, again, a function of the fluence. It's a function of the heat capacity of the material. It's a function of the thermal expansion coefficient. And it's a function of the optical absorbance. And so if you're controlling the fluence the same, and you're controlling the absorption or the optical density and hold that the same, probably, I suspect the difference between your different semiconductors is going to be a function of the thermal expansion coefficient or the specific heat. So, and that would make complete sense, right? Like different materials have different thermal expansion coefficients and they have different specific heats.
Right, exactly. So we have a couple questions specifically related to the dental applications. Can you explain a little bit more about how you're making a contact between the ultrasound and photoacoustic laser transducer with the actual teeth?
Yeah, so I was hoping to save this for a nicer paper, but I'm actually just writing a paper on it now because we're all closed. So this has a sleeve basically, right? We're not putting this probe directly on the teeth, but we've looked at a variety of different sleeves. We've looked at a variety of different conformations. But this is standard in the ultrasound, in clinical ultrasound. People put these sleeves on ultrasound transducers and then dispose them. It's basically like a condom. And so the real trick is just minimizing the air bubbles that are in that space between the transducer and the sleeve that then goes on the tooth.
Can you comment as to what the source of the photoacoustic signal is in the stain on the tooth's actual surface rather than in the pocket?
I don't, yeah, so clinically they call this calculus is what it actually is called. And I don't know. I don't know of what is generating that signal. I do know that it's routine in swine. And so I also don't definitively feel comfortable calling it calculus versus background staining. But yeah, this is of interest.
Yeah, great, thank you. We have a question. Do you foresee an opportunity to assess energy metabolism and its dynamics, i.e. the changing ATP versus ADP turnover in a mouse or rat model of myocardial infarction and reperfusion?
That's tough because you've got to have an absorber. So if you had, I would think that measuring free glucose is so established. If you had an absorber that was specific for ATP or ADP, and it was homogenously perfused throughout your target tissue, then yes, this would definitely work. I don't know what that absorber would be. And then just also be aware, of course, that FDG PET is a really great molecule that measures metabolic activity, and it's very quantitative. So fluorodeoxyglucose, radio-labeled sugar, inject that, do positron emission tomography, and there's your map of metabolism. So.
Yeah, great, thank you. Someone's referring specifically to the gangrene wound detection. It seems to show the images as a combination of photoacoustic and acoustic imaging. Do you know what the axial resolution of that is?
Off the top of my head, I don't, but it's hundreds of microns. I think this was done with a 40 megahertz transducer. This is on VisualSonics spec sheet, the exact lateral and axial resolution. If you write your sales rep, I'm sure they'll send you the spec sheet. But I just can't remember it off the top of my head.
Yeah, and unfortunately that question came in from an anonymous attendee. If you would like to add your name in, whoever submitted that question, we'll be happy at VisualSonics to actually reply back to you with a response to that. So we have a question, it's more of a basic question, but can you measure organ-specific oxygenation levels with this photoacoustic technique?
Sure, yeah, as long as they're not too deep. Your depth is going to be critical here. But sure, I mean, that's what's great about this technology is if you think about a pulse oximeter, you're passing light through tissue, through a finger, through an ear, through a toe. With this, all you do is wherever you point that transducer, you're getting a map of hemoglobin to deoxyhemoglobin. So if you want to do this experiment, I always like putting the animal on different amounts of oxygen just to convince myself that it's working. The other thing is these numbers never match perfectly. So if you look at the pulse oximeter number here versus the value I calculate here, This is because of differences in how these two different wavelengths of light propagate through tissue, among other problems. But it's very rare for these numbers to match exactly. Oops. So yeah, I mean, if you want to put this and try to do it in a human heart, I would say no, but to potentially do this in a mouse liver, absolutely.
Great, fantastic. And we're just rounding out at the top of the hour here. We do have several other questions. We will submit these questions to Jesse for a response, or the company VSI itself will answer back to you. So I'd just like to remind everyone, thank you very much for coming today. We really appreciate you taking the time. We hope, of course, that you're being safe. There are multiple resources on the VisualSonics website, such as past webinars that we have recorded. We have our Learning Hub. There are a variety of resources for upcoming grant deadlines. So please feel free to look at our website and, of course, contact any of our staff with any questions. We'd be more than happy to help you. So again, I just thank Dr. Jokers for your amazing presentation today. And with that, we'll conclude this webinar. Thank you.
Thanks so much.